It's 2 o'clock, so I think we should get started. I want to welcome everyone to the OCB 2020 virtual webinar series. For those who don't know me, my name's Heather Benway. I'm the Executive Officer of OCB in the OCB Project Office. I'd like to introduce my co-host, May Mahegan, who is the Communications Officer, Administrative Associate, Mary Zawoski. And there are 2 OCB Scientific Steering Committee members who are co-chairing our webinar today. We have Victoria Coles from University of Maryland Center for Environmental Science and Amy Moss from Bermuda Institute of Ocean Sciences, and they will be introducing our speakers today. The OCB office is based at Woods Hole Oceanographic Institution, and we receive generous support from NSF and NASA. OCB is essentially a network of scientists. We work across disciplines, a lot of chemical and biological oceanography to understand the ocean's role in the global carbon cycle and how marine ecosystems and biogeochemical cycles are responding to environmental change. We are involved in looking at biophysical interactions. There's just a very broad, swath of oceanography disciplines involved in OCB. You can look at our encompassing science areas here in these green icons, so you can see the science the overarching science areas and current. If you want to look at our current research priorities and questions, you can visit the URL at the top of the page. It's just the About page of our main website. Given the cancellation of our big annual summer workshop that we usually have in June in Woods Hole, we had to cancel that this year because of COVID. We wanted to find a way to keep our community connected and continue to share great new science. So we solicited ideas and abstracts from the OCB community a couple months ago, and then we went through the submissions to put this webinar series together. So we're really excited to be able to have this virtual presence. I want to also let you know that given the success and the attendance on these webinars, we do plan to continue them into the fall. And there's actually, you can go to this website and there's a link to an application to submit new titles and abstracts for new talks for the webinar series. So we invite you to do that. That application is open again. Here's a little teaser on a couple of the next couple webinars that are coming up. You can find the full list on the website. We share these on this website, and we also share these on Twitter and via our e-newsletter. Plenty of ways to engage with the OCB program and get involved. Please tweet. You can live tweet during the webinars using the hashtag listed. You can go to our website, subscribe to the newsletter to get plenty of detailed information about what our program is doing and what the broader ocean carbon community is involved in. And we do plan to post a recording of today's webinar. All of the webinars actually are on YouTube, on our YouTube channel. If you have any questions, you can always reach us by email at the address listed. Just a few guidelines. All of the attendees are muted today, as is typical for webinars, so that we can reduce background interference. We ask that you save your questions for after the talk. You can type them in during the talk, but they won't be addressed until after the talk. We are using the Q&A function for attendees to enter their questions. The moderator will go through as many as we have time for, and then any questions that we don't get to, we will send those to the speaker along with the person who's asked them so they can follow up with you after. If you have any technical issues, you can use the chat function, just chat with the hosts. And here's the dial-in information. If you find that your bandwidth is challenged, you can certainly just call in this number and listen in and then view the webinar later. Just as a little screenshot of what you're probably seeing right now. You can mute yourself. Actually, all the attendees are already muted, but you can access the chat here in green using this little bubble icon, and then the Q&A is available under this ... this more options. So you can pose questions that way. If you want to change your visual layout, it's in the upper right corner. And then the details for calling in, if you have to switch to the phone, are listed right under here. The theme of today's webinar is Advancing Understanding of Biological Feedbacks on Ocean Biogeochemistry, and we're really excited to have had so many people register for today. We were over 150 when I started the webinar, and I still see more coming in on my email, so I'm really excited to see that. Today's speakers are Emily Zakem from the University of Southern California and Severine Martini from the Mediterranean Institute of Oceanography. I'm going to let Victoria introduce our first speaker, Emily Zakem, while we transfer the speaker role to Emily.
Sorry, I'm having trouble unmuting. Hi, I'm having a little trouble unmuting.
We can hear you, Victoria.
Thank you. It's really my pleasure to introduce Emily. I've been following her work with a lot of interest since she was a graduate student because it's exciting and creative, as you'll see. Emily was a fine arts major at the University of Michigan as an undergraduate, which I think is a really nice example of how a diversity of backgrounds can give you a creative research perspective. She got her master's degree in interdisciplinary environmental studies at University of Colorado, and then went on to do a PhD in climate physics and chemistry at MIT, working with Nick Follows, who many of you know as a frequent attendee at our OCB meetings. She's now a postdoc at the University of Southern California, working with Naomi Levine and funded by the Simons Foundation Postdoctoral Fellowship in Microbial Ecology. And for those of you that want to know just one more thing about Emily for your question and answer, she tells me that she learned to sail while working as a camp counselor. And apparently that was so enticing that she ended up living on a sailboat in the Great Lakes without running water or electricity during her period there. So take it away, Emily.
Okay, thank you. Just checking, can you hear me right now? Okay, great. All right.
Yep.
Okay, so I'm going to talk about modeling, and specifically I'm going to talk about an approach to modeling that I've been working on. So just starting from the real basics, if we're just talking about the Earth system as a whole, we can boil down biological processes to two main processes: primary production, or the synthesis of organic matter from inorganic nutrients by phytoplankton, and remineralization, or the return of that organic matter back into its inorganic constituents. Both of these processes we know happen in the sunlit layer of the ocean, but because of the gravitational sinking of organic matter, the processes become decoupled. And that decoupling is what forms the biological pump, or what we call the biological pump of carbon in the ocean. So here I'm just showing you nitrate concentrations in the deep ocean. We know that there's a proportional increase in the concentrations of dissolved inorganic carbon as well. And so without this biological pump, the atmospheric CO2 would be about 100 ppm higher than it is. So we know that respiration at depth is important for the climate and the Earth system. But it's not just photosynthesis and respiration. So here's a sampling of 12 metabolisms, all carried out by microbes, except for photosynthesis and respiration, which are also carried out by larger organisms and such. And so these 12 are just examples of metabolisms that are biogeochemically important in the Earth system. And here I'm just representing them by the redox reaction for each of them. And we know that many of these are important for setting concentrations of atmospheric gases, which I've indicated the ones that are greenhouse gases in red at the bottom of the slide here. So the ultimate question is, how do we connect these processes? How can we describe these metabolisms, mostly carried out by microorganisms, mechanistically, so that we can understand the feedbacks to the climate system? So how do we connect these scales? So global ecosystem models become very useful tools. They let us synthesize, untangle, and quantify these complex interconnected processes. So historically, there's been a very disproportionate way in which these microorganisms have been represented in ecosystem models. There's been typically a lot of attention to the primary producers, the phytoplankton. Most climate models have half a dozen phytoplankton functional types as state variables in the models. Zooplankton are often included as well. But the return reaction of that primary production, the remineralization, has often been represented very crudely or very simply, usually with first order rate constants, rather than resolving the biomass of the organisms that are carrying out these processes. This is due to, there's a lot of reasons for this. And this is changing very rapidly right now. We have so much information about microbial metabolisms other than phytoplankton, and models are incorporating these diverse metabolisms. So one example is Victoria Coles and co-authors. They developed a model of diverse microbial metabolic functions, and they focus particularly on the interface between modeled quantities and the metagenomic and metatranscriptomic information, which is really the information that we have to understand microbial metabolisms in the ocean. So as we progress towards these new models with microbes, what is the best way to incorporate metabolic diversity? So what is the goal? So there's not one best way for all questions, but what is a meaningful way for climate models, for example, where we really want to understand feedbacks? So the goal for these sorts of models is we want to be able to make predictions about biogeochemistry in unobserved environments, like the past and future oceans. So what's not useful for these sorts of models are descriptions of metabolisms that are specific to a site or specific for a species. What's also not useful is to have many unconstrained parameters, because that really increases model uncertainty. Is helpful, what we ideally want are universally applicable descriptions that are relevant for a wide diversity of environments. And ideally, we want to minimize additional degrees of freedom introduced into the model as we introduce more and more metabolisms. So today I'm going to talk about one approach that works towards this ideal, where we can use the redox chemistry that underlies each of these metabolisms in order to quantitatively link the fluxes of substrates to biomass in meaningful ways. So here's just a super simple cartoon of a cell. And so in this simplified framework, you can think about the required substrates for uptake. You can think about which of those substrates are used for biomass and which are used for respiration, and which substrates are in modified form due to that respiration. And there's a methodology that was originally developed in wastewater treatment plants to organize these relative fluxes using the free energy that's released from the redox reaction that fuels the cell. And this allows us to quantify the relative fluxes here and understand them in an environmental setting. So as a more specific example, this is a more complex version of that cartoon for an exemplary metabolic type, which is what we can call them. So for an ammonia oxidizer, for example, we can understand its required substrates, oxygen versus ammonia and CO2, and relate those to biomass synthesis versus respiration to form nitrite. So for example, it quantifies the amount of ammonium taken up by the cell relative to the amount of synthesis that can be fueled by the oxidation of that ammonia. So I'm going to talk about the benefits of this type more specifically by using two examples from research. So the first example is about nitrification. So nitrification is the two-step process by which ammonia is oxidized to nitrite and then to nitrate, filling the deep ocean. And we know this is intertwined with the biological pump because the places in the ocean below the euphotic zone where nitrification is maximized are it's the same location where we usually measure export production. So the question is, why is nitrification peak below the euphotic zone at depth? So here's some observations from Allison Santoro in the Leucotropic Pacific Ocean. So on the left, there's abundances of ammonia oxidizer and nitrite oxidizer microorganisms, as well as abundances of chlorophyll A as a proxy for phytoplankton. And on the right are measurements of ammonia oxidation rate. You can see that both the abundances and the rates of nitrification peak at depth. And there's this systematic difference in the abundance of the ammonia oxidizers versus the nitrite oxidizers. So why is this? So about these metabolic functional types of these two nitrifying populations by relating that redox chemistry to biomass synthesis. And here's the end of that. Here's a whole organism simplified budget of the metabolism. So this methodology is sort of like a very coarse-grained flux model. And so this articulates two things here that are important. First, that the yields of biomass with respect to the inorganic nitrogen taken up are very low. So for both of these, you need at least 100 moles of ammonia nitrite to produce 1 mole of biomass nitrogen. So it's overall very inefficient. Second, the redox chemistry suggests that there's approximately a 3-fold difference in the yield, that you need about 3 times more nitrite relative to ammonia to produce the same amount of biomass. So we can develop these metabolic functional types and then incorporate them into an ecosystem model. So here, these types are part of a larger ecosystem we already have models that have these descriptions of zooplankton, phytoplankton, organic matter, etc. So here I'm just going to show you an incorporation dimensional water column model where there's euphotic zone, there's mixing, a mixed layer, and a sinking of particulate organic nitrogen from the surface. And so most models that include nitrification have some sort of inhibition of nitrification as a function of light. So the exclusion from the surface layer is imposed. I'm going to show you how using these metabolic functional types lets you understand the emergent biogeochemistry without prescribing light inhibition. So these are the solutions. We have the model here has excluded nitrification and most of the abundances from the surface of the ocean. And this is due to the interaction between the phytoplankton and the chemoautotrophic nitrifiers. The phytoplankton are much more efficient at converting inorganic nitrogen into biomass, and so they competitively exclude the nitrifiers from the euphotic zone. So this lets us understand the biogeography of nitrification more fundamentally. So also the model distinguishes, it's able to capture the difference in the abundances. So this difference in abundance reflects the redox chemistry as well as any differences in cell size. So that's useful as well because it lets us relate the modeled quantities to others. And also what's useful is, so this simplified flux balance model links together elemental cycles. So we have estimates of how much carbon fixation is happening due to these chemoautotrophic metabolisms. So here's the global configuration of that same model. I'm just going to show you it very quickly, but in the top, there's the model and data for nitrite concentrations, which are concurrent with peak nitrification rate. And the bottom is an estimate of the surface nitrification rate. And this is consistent with some observations that show that nitrification does indeed happen in the surface. And so we can come to a more general hypothesis and a more general description of nitrification for all environments that shows us that nitrification becomes energetically favorable whenever it is that photoautotrophy is limited by light. So that can be below the euphotic zone, or it could be at high latitudes in the wintertime, as this global configuration suggests. So what about that light inhibition? We know that real microorganisms are inhibited by light. So here, by linking metabolisms to the underlying chemical potential, we can say that, well, maybe light inhibition evolved as a consequence of long-term exclusion of this metabolism from the surface because it's just not energetically favorable there. Okay, second example is to do with oxygen minimum zones in the ocean. So what governs the transition from aerobic to anaerobic metabolisms? That's an interesting question for us. So we know that oxygen minimum zones are formed due to the relative supply of organic matter and oxygen at depth. And when oxygen becomes sufficiently depleted, microbes turn to anaerobic metabolisms for energy, and this leads to the losses of fixed nitrogen from these zones. So we're interested in how nitrogen loss from the ocean, denitrification and anomox, how that might change in the future. So we can employ our metabolic functional type approach, and OMZ biogeochemistry is super complicated in an exciting way. We need at least 6 metabolisms to capture the nitrogen and oxygen carbon cycling that are happening there. Most models of OMZs, even ones that do resolve the biomass, employ some sort of prescribed oxygen concentration that determines the presence or absence of each metabolism. And here, what's useful with using this redox methodology is that we don't have to rely on those arbitrary empirical thresholds. We can allow these energetically described metabolic functional types to interact with each other. So we'll take these 6 metabolisms and I'll show you what happens when we integrate them into a super simplified flow field to form an idealized oxygen minimum zone. So this is just the flow field with a passive tracer showing you this spin up of a passive tracer, just illustrates the circulation. So this 2-dimensional flow field resolves oxygen surface as a consequence of that, intensified upwelling along the coast and dispersed downwelling. And this is what we need to get the essence of oxygen minimum zone dynamics. So if I integrate those 6 metabolic functional types into this model, along with phytoplankton and all our DIN tracers and seeking organic matter, et cetera, and run it to steady state, this is what we get. So on the left are the observed and modeled oxygen concentration. And on the right are those 6 metabolic functional types, just the biomass that comes out from the model at a steady state. So you can see that without prescribing oxygen thresholds, we've been able to understand how and why aerobic metabolisms become energetically favorable in oxygenated waters and anaerobic metabolisms become favorable in anoxic waters. This is due to the ecological interactions between these types. And this is useful because it sorts out the relative abundances of the biomass associated with metabolisms in a meaningful way. So for example, we know that anomox contributes to roughly 30% of nitrogen loss on average. And this model is able to capture that 30% ratio and also capture the observed very low abundance of the anomox organisms. So this is useful because it makes the point the abundance of an organism doesn't necessarily have anything to do with its biogeochemical significance. And linking redox chemistry to those abundances is a way to make a prediction about how that decoupling happens. So that's it. I'm going to summarize those benefits really quickly. On the right is just a quick snapshot of showing what these metabolic functional types look like in a model from the nitrification simulation in a global model using the Darwin MIT JCM framework. So how much do I have left here? Okay, so the main benefit is that we can have metabolic biogeochemistry emerge in our microbial ecosystem models, and that allows us to understand it more fundamentally and make more meaningful predictions in different environments. We don't have to rely on imposed thresholds to determine the presence or absence of these different metabolisms. Allows us to incorporate feedback from all the remineralization processes, which is it articulates and quantifies relationships between abundances, rates, and concentrations for different metabolisms. That's useful. And because it resolves functional biomass, it lets us compare model results to sequencing data sets more easily. Limitations, there's a lot of limitations. So anytime you add a state variable to a model, it becomes more computationally expensive. So this may be useful, this approach may be useful for some questions, but maybe not for others. It doesn't inform everything. It doesn't give you all of the answers. So it doesn't tell you how metabolisms are distributed among a population. We have to make that choice. There's nothing here that's said about the physical processes such as particle dynamics, the sinking rates of particles or niches inside particles. There's a lot of complexity to organic matter and the heterotrophic microbial community that is not simply predicted by this redox approach. We can use it as a tool to start digging into that, but it's not clear-cut way forward there, and there's a lot of complexity that's important for understanding the biological pump there. And of course, loss rates are always critical for predictions about microbial any sort of population in our ecosystem models, and these still remain largely unconstrained. So that's a huge uncertainty in these models. So all this we've written into a perspective. I put this together into a perspective written with Martin Poltz and Nick Fallows. And with that, I'd just like to thank my co-authors, Mick and Martin, on this project, as well as everyone past and present that's worked with me on these projects. And a special shout out to Heather and May for helping set up these talks. It was just really fun to work with them. And the Simons Foundation for funding the research. So thank you.
Thank you so much, Emily. That was fantastic. I'd like to open it up for questions right now. If people have questions, just a reminder, you can use the Q&A function, or you can use the chat if that's more available to you. The chat is the little speech bubble, and the Q&A is under the ...
So while we're waiting for people to, get their questions in, I'm anxious to get mine in first. So you guys can find that Q&A function. Thank you, Emily. That was really exciting. And I think that was a really beautiful illustration of how these processes that sometimes we prescribe, like light inhibition and nitrification, might not emerge everywhere. So I was curious about your global model. When you include that process of nitrification occurring at the surface in the polar and subpolar regions, what does that do for your carbon budget and your export?
Oh, that's a good question. Yeah. Well, it turns out that these chemoautotrophic nitrifiers they're operating at extremely low abundance. So in terms of carbon, I know this is OCB, but they don't seem to have a huge impact on the carbon cycling in terms of export production. I have to think about that. Maybe there's something that I'm missing there. There's certain
but they suck up all the nitrogen.
Yeah, so that's what's interesting. They don't suck up all of the nitrogen. There's still nitrite there and ammonia there. In the model, that's because of the turbulent mixing at depth. And yeah, so I think there's a lot of really open questions there in terms of what the biogeochemical impact is, but it does seem that the nitrifiers, for the most part, in terms of the carbon budget, are sort of in the background. The carbon fixation rates are very low, but that's a big open question right now, is what are those carbon fixation rates globally for the chemoautotrophic nitrifiers?
I see that Julie Granger has a question. I'm going to try to unmute her so that she can ask it herself. Julie, can you hear us?
Yeah. Can you hear me?
Yes.
Okay. That was a wonderful talk. And I have two small questions, or maybe two big questions. My first one is, do you impose a death term, say, on the nitrifiers to control their biomass? I'm supposing you have to. And then my second question is, within the OMZs, say there were metabolism that you were prescribing that was actually there and could be important, could your results look like they fit the biogeochemistry but actually are still missing an important flux? And I'm thinking now of like whispers of anaerobic nitride oxidation, which may or may not be happening and may or may not be linked to the carbon in therein.
Yeah, great questions. First, yes, there's loss processes, dynamic loss processes for the descriptions for all the populations. The way that I did it in my model, I assumed that there's two parts to the loss. There's a black box viral component. We assume that there's losses to viral lysis that I just parameterized using a quadratic mortality constant. That's what we do. And then I also included a dynamic zooplankton population. So we can imagine there might be some very small zooplankton grazing on bacteria. They're super uncertain. The way that I dealt with that uncertainty in the model is that I assumed that those loss forces were the same for all the populations. So they weren't determining the outcome of the solutions. And the uncertainties in the loss processes didn't change my results qualitatively. As for the OMZs, if there's other metabolisms that are missing, yes, I think there absolutely could be. I think there's different ways of doing models. There's so many different ways of doing models. The way that I use them is I use them to understand mechanisms. And so I'm not too concerned with always matching the numbers precisely. So that means in my model simulations, there's a lot of wiggle room for understanding how there might be other metabolisms taking place. I don't resolve the sulfur cycle, for example. I'm not particularly concerned so far in my work of simulating the exact nitrite concentration of the secondary nitrite maximum, for example. So yes, models are only ever as good as the assumptions that you put into them, but I think there's ways to use the simplified versions of the biogeochemistry to answer some questions, but not others.
I don't see any more questions. So I think we will move on to our next speaker. Thank you so much, Emily. We will, if any questions do pop up, and anybody who has any can feel free to put them in the Q&A, and we can send them on to Emily to address later. So as we transition speakers, I'm going to let Amy introduce our second speaker, Severine Martini.
So I'm really excited about this next talk, and I've noticed a theme. There's a theme of living on sailboats that appears to be part of our session. So Severine grew up landlocked in Lyon, France, but she had always been sort of fascinated by the sea. So when she was 15, she went sailing for a year. And she said that the most striking memories she has are the luminous patterns behind the sailing boat, which is very fitting for what she ends up doing. At that point, she pretty much knew nothing about bioluminescence, but she said it reminded her of the stars in the sky. Severine then did her PhD at the Mediterranean Institute of Oceanography in Marseille, France. And again, there was this linkage between the stars and the sea, because although she was working on an oceanographic data set, it came directly from a neutrino telescope, which had at first been dedicated to astrophysics. So these things, they come back around. And during her degree, she became really focused on bioluminescent organisms, including bacteria. And after the completion of her PhD, she took a postdoc at Monterey Bay Aquarium Research Institute in California. And she's going to be presenting some of the work that she did there as part of this. And she's now back at the MIO in France, still working on glowing critters, and is now trying to develop new instruments and methodology. And I hope you enjoyed this talk as much as I did when I saw it the first time around. So Severine, please.
So, hi everybody, my name is Severine Martini and I'm a researcher at the Mediterranean Institute of Oceanography. And today I will shed light, literally, on processes within the biological carbon pump. Literally because my interest is to understand if and how the light emission by organisms impact the carbon sequestration in the ocean. As a brief summary, you all know, of course, that the biological carbon pump is a major process regulating the atmospheric CO2 through trophic interactions between organisms. And from this interaction, the particle flux sinks to the bottom of the ocean through the meso and bathypelagic zones. But if this carbon is produced at the surface of the ocean, it is transformed in the deep, dark ocean. And darkness constitutes the main feature of the ocean during the night, even the ocean surface is dark. The deep, dark ocean represents actually 94% of the Earth's volume, so it's something that is very important on Earth. And the dark ocean is also characterized by a high pressure and a decrease in temperature. And within this environment, a lot of organisms have the capability to emit their own light, and this is called bioluminescence emission. Bioluminescence actually is a functional trait of organisms, and the functional trait is a common characteristics shared between different taxonomic groups. While bioluminescence is something that is very rare on Earth, on the terrestrial land, actually it's something that is very common in the marine environment. And there are pelagic organisms as well as benthic ones that are able to emit their own light. Bioluminescence is actually mainly emitted in the blue wavelengths, blue-green wavelengths, but there are some exceptions, like for example, the Tomopteris worm on the right of the screen here. These organisms emit some yellow bioluminescence, which is something that is very uncommon compared to the bluish bioluminescence that you can see on the left of the screen here. In a precedent work, I've been working on a very unique database, which is available at MBARI, and this database is called the VARS dataset. And I used observations that have been recorded during 240 ROV dives from 100 meters depth down to 4,000 meters depth. And those observations have been mainly performed in the Monterey Bay in California here. And those observations include about 17 years from 1999 to 2016. And it's about 350,000 observations. So it's a very unique data set. And thanks to this unique data set, what we've been able to do is that we've merged those observations with some knowledge from the literature about the capability of emitting light from those organisms. And we quantified the percentage of bioluminescent creatures observed from the surface down to the deep. This is what you can see on the graph here. This graph represents the depth from 0 about the surface down to 4,000 meters depth. And then the percentage of bioluminescent organisms in blue, dark blue and light blue, non-bioluminescent organisms in dark gray and gray, and then the undefined capability of organisms for bioluminescence emission is represented in yellow here. And something that is very interesting on this graph is that there is actually about 76% of organisms for all depth, and 76% of organisms that are able to emit bioluminescence. It's pretty constant among the depth. And something that was even more surprising is that while we know pretty much the bioluminescence capability of those organisms above 500 meters depth, so there is almost no yellow colors, the percentage in yellow of unknown is very low. However, below 500 meters depth, this percentage increases. The yellow percentage represented here increases with depth. So that was some very interesting result from this graph. And this means that when you are going deeper and deeper, then there are still a lot of things to learn. And to summarize these results, so what we found is that there is 76% of pelagic organisms that are known to be bioluminescent, And we've done a very similar study in the same observations, but for benthic organisms. And what we found was that there is a lower percentage of bioluminescent organisms, more likely to be 30 to 40% of benthic organisms that are known to be bioluminescent. And then what is represented here, 24 and 60, are the percentage of organisms that we have no idea if they are luminous or not. So emitting light in the complete darkness is an effective way to escape a predator, for example, which is the defense category. Bioluminescence emission is also used to attract a prey, for example, which is the offense category. But it's also very useful in a complete darkness to find a mate for reproduction. So those are different ecologic roles of bioluminescence emission. And most creatures emit their own light, but some benefit from a little help, because some organisms are not emitting bioluminescence by themselves, but they are in symbiosis with some bioluminescent bacteria, actually. And one very well-known example is the anglerfish, which is a fish which is using a lure full of bioluminescent bacteria to attract prey and feed on them. So this one is very well known, of course. So there are a lot of very different bioluminescent organisms and, of course, bacteria. And those bioluminescent bacteria can be found either free living in the ocean or in symbiotic association, but also attached to particles, sinking particles of carbon. The interest for those bacteria is that when they are consumed by fish, for example, they're going to find a better environment to grow with nutrients, for example, and the temperature is also different. So they're going to grow in a better environment than when they are free living in the ocean. And for the host, of course, the benefit is that bacteria provide light and they can use this light to attract prey. Interestingly, in the literature, what we found is that it has been shown that about 70 to 98% of particles have been observed to emit light, to be bioluminescent. And those particles were either fecal pellets or particles collected from sediment traps. And the glow that was observed on those particles was mainly due to microorganisms. And actually, some of the bacteria are attached to sinking particles, but those luminous bacteria can also be attached to suspended particles, for example, and they can also be found in fecal pellets excreted by fish or other organisms. Another fact from the literature, which is very interesting, is that numerous organisms living in this dark environment, have a vision which is well adapted to detect bioluminescence. They are able to detect a very faint light, very low light, within the wavelengths emitted by those bioluminescent organisms, bluish colors. And they are able to detect this bioluminescence, this light emission from a very large distance, several meters away. So the bioluminescence emission could certainly interfere and have an impact on mesopelagic trophic networks. A different study has also highlighted that the attraction of bioluminescence, to biological light, can have an effect on trophic levels. And this is called the bait hypothesis. And this article, for example, has shown this, well, they have kind of proved this bait hypothesis. And bioluminescence is visually detected and is going to act as a lure to attract zooplankton and fish. And in this study, actually, the authors demonstrated that at first, the glowing bacteria can attract zooplankton, and this is going to be twice more efficient than non-glowing bacteria. So luminous bacteria are going to attract more zooplankton than non-glowing ones. Then the second step is that when the bacteria have been ingested, their lights can be observed within the zooplankton guts. So this is the second step. Then the third step is that the attraction of fish is also 8 times more efficient for zooplankton, which have been consuming luminous bacteria compared to zooplankton, which have not been consuming those luminous bacteria. So a non-glowing zooplankton is going to attract less than a glowing one. And this is the third step of the bait hypothesis. And finally, something that is also very interesting is that even though the luminous bacteria have been ingested by zooplankton and then by fish, for example, when those fish are excreting fecal pellets, those fecal pellets are still glowing, which means that the luminous bacteria are still living and active in the fecal pellets after being in the guts of the zooplankton and then the guts of the fish. What you can see on this picture, on the left, those are fecal pellets under white light, and on the right, those are fecal pellets glowing due to luminous bacteria. One of our scientific question based on all of this literature is what could be the impact of bioluminescence and bioluminescent bacteria on the biological carbon pump? And we reviewed the literature to answer this question. So all the literature that has been published on bioluminescence and potentially how it impacts the biological carbon pump. We reviewed all this literature and we published recently a review called Bacterial Bioluminescence, Ecology and Impact in the Biological Carbon Pump with some colleagues in Marseille. We tried to summarize the global effect of bioluminescence in the biological carbon pump by some hypothesis that we call the bioluminescence shunt hypothesis. And this can be summarized with this representation, because the bioluminescence shunt hypothesis proposes bioluminescence as a catalyst and amplifier that could influence the BCP efficiency. So from the beginning, the particles are easier to detect due to the visual tag from the bioluminescent bacteria. And this detection will increase the consumption of those luminous particles by zooplankton and higher trophic levels. Then this can have 2 different consequences. The first consequence is that the hypothesis is that such consumption will increase aggregation in the guts of the fish, for example, followed by excretion of fecal pellets. And those fecal pellets, the glowing ones represented here, are more dense and with a higher sinking rate than the particle at the beginning. So this aggregation in the guts is going to increase the sinking speed and at the end the carbon sequestration in the deep ocean. On the opposite, the second hypothesis is that the consumption of those luminous particles can increase the fragmentation of the organic matter. So when zooplankton or fish is consuming those particles, then they're going to fragment the particles, which are going to be less dense with a lower sinking rate. And this is going to decrease the carbon sequestration at the end. So what we propose is 2 different hypotheses, the aggregation and the fragmentation, based on the impact of bioluminescence in the BCP. Currently, what is known is that the particle degradation by higher trophic levels is strongly influenced by the chemical plume detection. And this can impact zooplankton detection at a distance of few centimeters. And what we propose within this bioluminescence shunt hypothesis is something that is similar to the chemical plume, but related to the visual attraction. And what we found interesting is that while the chemical plumes can have an effect a few centimeters away, the visual detection can be can impact the higher trophic levels up to several meters away. So the detection can, the visual detection can happen from several meters away. In our global representation of the bioluminescence shunt hypothesis, luminous bacteria can be released by a symbiotic association, for example, a symbiotic squid, or like a squid or a fish with symbiotic association with bacteria can release bacteria into the water on fecal pellets, for example, or free living in the water. And those can be the sources of luminous bacteria. Then the second step is that the light emission on fecal pellets or free-living bacteria that are going to be attached to particles can increases the detection and consumption rates by higher trophic levels. So this is the second step. Then the first step is that there can be two opposite consequences in this consumption. The first one is that it can increases, and the second one is that it can decreases the particles sinking rate, and at the end, the carbon sequestration in the deep ocean. Then the fourth step is that the vertical migration can also allow the luminous bacteria to be more dispersed through vertical migrations. And the last step is that when sediments are resuspended, then this can also act as a physical resuspension of luminous bacteria. And then the luminous bacteria will be back into the loop by being seen and consumed again by higher trophic levels. In order to test those hypotheses, the bioluminescence shunt hypothesis, in our team, so in the review that I presented, we propose a general pathway for the future. So some different actions that can be that can allow to check the bioluminescence shunt hypothesis. And in our team, we think that it is necessary as a first step to better understand bioluminescence variability in the ocean at the general scale, at the global scale. For example, we developed a very small tag able to record bioluminescence in the pelagic environment, and this tag is called sensor, for high sensitivity sensor for bioluminescence detection in the dark ocean. And it has been first deployed on elephant seals, so we got some first data in the dark ocean. But more generally, there are some oceanographic instrumentation that are already available for future investigations on the bioluminescence shunt hypothesis. And to describe the diversity of luminous bacteria, for example, the ones that are attached to particles, there are some tools that are commonly used in biogeochemistry. And for example, using sediment trap, it is easy to sample particles and then to look at different species of bacteria that are attached on particles. Those particles can be observed, and then the bioluminescence of those particles can be quantified. And it is important to quantify the number of luminous particles to see if there is some spatiotemporal variability of these luminous particles. So, yeah, spatiotemporal variability of light emission on particles, but also generally in the global ocean is important. So there are some tools that have been deployed, not so much actually. So we need a general effort on having a better knowledge of spatiotemporal variability of bioluminescence and light emission in the ocean. We also need to observe the attraction due to light emission in predator-prey relationship. So how the light emission by bioluminescent bacteria is going to attract higher trophic levels. So this can be done in situ, but this can also be done in the laboratory. So all those different tools are going to allow us to test this bioluminescent shunt hypothesis, and at the end, it's also going to be an important step to quantify the potential impact of bioluminescence in the BCP, the biological carbon bond. And I think this is something that is quite new. Yeah, if you want to read a little bit more, then the review that we just published is provides some very good background on bioluminescence and how it can impact the BCP. So thank you for your attention.
Thank you, Severine. All right. I think we're at the place where everybody's going to start figuring out where their chats are and what kind of questions they want to ask. And so I'm going to do what Victoria did and ask my question before anybody else can get to it. So I love this, starting to look at how the ecological interactions are dictated by these light things. But because I always am thinking in the context of climate change and the exports of things, what do you think as things warm and stratification changes, are there any sort of implications for how this this functional trait might change in a changing climate and what those implications could potentially be?
I think the question that we have to, so the first question that we have to answer.
Can you speak up a little? We can't hear you.
Okay, sorry. Is it better now? Okay.
Yes.
So the first question that we have to answer is either if this bioluminescence shunt hypothesis is going to increase the carbon sequestration or decrease it. And those are so it can be very different consequences actually. So for now, we don't know if the bioluminescence is going to increase the rates of aggregation or fragmentation, for example. And there is a recent publication from Nathan Briggs at the beginning of the year that has been showing that fragmentation is something that is very important, but they couldn't describe the reasons for this fragmentation. And I think it's very important to test and take into account the bioluminescence because it's something that is widely represented in the ocean. And maybe if bioluminescence is a big part of fragmentation, then yeah, I think it can definitely change the carbon sequestration.
Yeah, I was wondering whether perhaps bioluminescent colonization of particles would increase with warming, just as the physiology of them increased. And then the downstream stuff, we don't know, right? Whether the fragmentation or the aggregation would be more prevalent, but yeah. Thanks.
Looks like we have one question in the Q&A. How do bioluminescence and low oxygen zones correlate?
So for the bioluminescence emission, there is oxygen which is related to the chemical reaction, but I don't know actually how this is going to impact low oxygen zone.
Can I add a comment there? There's a graduate student who's been looking at how low oxygen affects the visual systems of zooplankton and fish. And so there's a potential interaction between these higher predators being able to see things under low oxygen. So that's a potential overlap that I've heard of.
There's a question here. Why do you think bioluminescence was so high in the trap particles at 30 meters in the study by Carl and colleagues?
In this one, I think at the lower depth there is, for example, there can be some dinoflagellates attached to particles. It's not only bacteria; it can be microorganisms and bacteria. So I think maybe in the upper part of the water column there can be a lot of dinoflagellates attached to particles. Yeah, sorry, and those dinoflagellates can be bioluminescent.
That's about all I see in the Q&A. There's a compliment on your figures and how visually stunning and they are and how well they communicate what you're.
So I have to say something about that if you appreciate the figures. So Lisa Tanay was a PhD student in my lab, and she just finished during the French lockdown. And she is the one that has been working a lot on making the figures beautiful. So I think she's the one that deserves all the comments, all the good comments.
Any other questions before we wrap up today? All right. Well, I want to thank our speakers again, and we will get this webinar recording up on our YouTube page as soon as possible, so you can share it with your colleagues. I want to thank everyone for coming today, and we look forward to seeing you on the next one.